Hello and welcome again. Well, it's been 1 year approximately since we at CIS, Canadian International School, entered into the lockdown due to the COVID-19 pandemic. Since that time, of course, we have returned to campus for a little bit of hybrid learning, but currently we are still very much in the midst of the pandemic, and actually the numbers are beginning to rise again in Bangalore. But despite that, we are celebrating the 25th anniversary of CIS this week, so happy birthday to CIS. And of course, we are moving ahead, despite the pandemic, to conduct our second major lab report for the school year. And this one, of course, is a full rehearsal for the final IA, which you will have to write up in IB2 sometime around the month of October for us at CIS. So let's move in and take a look at the research question. We're dealing with the topic of enzymes, and we are looking at the browning of plant tissue, specifically with respect to bananas. And how does refrigeration at 5 to 9 degrees Celsius affect the browning as measured by a colorimeter app of the banana Musa SP? Now, notice in this particular research question how I've made it focus by speaking about how I'm going to change my independent variable temperature, and also adding just a bit about how I'm going to measure my dependent variable, which is the change in color of the banana. Notice also that we are using a quantitative method to measure the color change. We're not just going to look and say the banana was light colored and it became a little bit more brown, but we are going to try to quantify it using this colorimeter app, which we can have on our devices. And of course, in keeping with the nature of science, we are following the lead of chemistry students over at the University of Southern California, who used a similar procedure when they carried out their labs in remote learning just about a year ago, and their teachers were able to publish the procedure. And of course, this is the nature of science, where one researcher publishes something, other researchers read it, and they can apply it to their own situation. So we are doing exactly that. We are going to learn how to use the red, green, and blue analysis system of the colorimeter to look and to track the color changes of our banana in one temperature versus another. So we're going to look at 5 to 9 degrees Celsius, and we're going to look at normal temperatures as well. So how does the refrigeration at 5 to 9 affect the browning of the banana? And of course, we'll be comparing it. So perhaps that too could be added to this research question. You can rephrase your research question as you see fit. I just wanted to use this as an example. But this particular lab was carried out many years ago by my students and I want to take you back to that discussion where we were talking about how this lab would happen when there were all the options available in the school lab. And to access that video, you can click on this card above if you have cards enabled, or you can click on the link down below in the caption to this video on YouTube, and you can access that. And then there's another video showing how I made the banana extract in the lab. But there are other ways to make it at home, and I'm going to show you how to do that. And I'm going to show you all of the materials that you need to use at home and how you're going to set this lab up so that you can do a meaningful scientific investigation and learn about a new way of statistical analysis, the t-test, as we look to do 10 trials of banana browning somewhere in this range, which is the reason I put that range is because your refrigerator is likely to be somewhere in between there, and you won't have a thermometer necessarily in there to tell you the temperature as well. If you do, of course, you can report that. So 5 to 9, that's the refrigeration in the lower compartment of most refrigerators. And then we'll be comparing that to the temperature that you might have in your kitchen in Bangalore, which might be somewhere between 25 to 30 degrees Celsius. Again, that temperature won't be an exact value unless you have a thermometer available, which some of you may and some of you may not. So let's move in now and take a closer look at some of the details to carry out this investigation. And here you can see some of the materials you would need for this lab. Of course, the more of these little containers that you have, the better it would be for you, because we would need to carry out 10 trials at each temperature. Here you crush the banana and you add some water to make a little bit of a banana extract. And then you go on to filter this extract and to collect it in these containers. Then you take a photo with your camera at a set position with the lighting controlled and a number of variables controlled to know the starting point of the colors. And you cap your containers and you put one into the refrigerator and you keep one on your kitchen counter. To use the color meter, you simply open your photo and you hover your mouse over a specific section of the sample. Then you leave the samples to undergo change for a set amount of time and you return to the same location with the same lighting and all of the variables controlled to take another reading, noting the red, green and blue readings once more.